I think we should start now because I can see a lot of people have already joined. So, okay. Hello everyone. My name is Sukhbir Ratan, and I'm the business manager at the Revity Signals for the India region. Thank you very much for joining this India Connect event today. Firstly, on the behalf of the entire Revity team, I welcome you all to this event. Thank you. Today, I am very excited to see this wonderful participation in this live Connect event, which is primarily focused on the Indian market. Today, we have a great lineup of the industry experts and our colleagues from the Revity who will share their insights and experience in data management, digital excellence, and collaboration within the pharmaceutical industry. I believe this event is a great opportunity to hear the best practices and ask the questions from the speakers. Moving ahead, I would like to introduce all my distinguished speakers for today. And to start with, my first speaker is Pierre Moriou, who leads the chemistry solutions at the Revity Signals. And his topic of presentation is data capture to decision making, harnessing the power of data management. Our second speaker is Ms. Rupali. She heads the digital excellence from the CIPLA, and her topic of presentation is optimizing compliance in the scientific research through electronic notebook, best practices and implementation strategies. Our third speaker for today is Tanvira Paul, who is a field application scientist at Revity Signals, and her topic of presentation is unlocking efficiencies, biosimilar research in Signal Notebook. Thereafter, we have a session from Mr. Ranga Rao, who is Associate Director in Bugworks Research, and his topic of presentation is transforming startup dynamics in pharmaceutical research, digitalization for accelerated innovation. And we will have a final session from Mr. Manish Kandagle, who is a senior field application scientist at Revity Signals, and his topic of presentation is accelerating formulation development with Signal Notebook. So we wanted to make this event very interactive, and therefore we would be starting this event with a quick poll to understand the who exactly are our audience. Asha, may I request if you can start the poll? So I request everyone to answer this poll. We will give 60 seconds to answer this poll. You can see, Arsha, can we have the answers of the poll? Okay, so we can see here there is a quite a diverse audience we have today, ranging from a formulation to chemical synthesis to biology and some from the IP and quality team also. Thank you very much for participating in this poll. Moving ahead, I would like to invite my first speaker for the today's session, Pierre Moriou, who is the Chemistry Product Marketing Manager at Revity Signals. Uh, Pierre basically help to translate the various chemistry from the time-saving discovery, enabling integrated software applications at Revity. Before to this role, Pierre has been a global product and marketing manager for the ChemDraw, which is a well-known tool, since 2017, where he has been overseeing the development and the launch of various version 17 through 21 of the ChemDraw and the ChemOffice suite. Pierre holds a chemical engineering degree and a PhD in the pharmaceutical sciences from University of North Carolina, and he has done his postdoc from a bioorganic chemistry also. So let me request Pierre to start the session. Thank you very much, Pierre. Over to you.
Thank you very much. Just making sure you can hear me correctly.
Yes, we can hear you correctly. Thank you.
And you can see my screen.
Yes, we can see your screen also.
Wonderful. Good afternoon, everyone. Thank you for joining us today. It's my pleasure to start the sessions of this India Connect. And what I would like to do is give you a very brief overview. I'm not going to take too long of Revity Signals. Maybe it's a name that you are or you are not familiar with. And very briefly, it's a new company. It's not even a year old yet. And really, the couple names that you need to be mindful of are ChemDraw, Signals, and Spotfire. So we are a software solution provider, a scientific software solution provider that empowers chemists, biologists, and decision makers to gain critical insights from any scientific research data with the overall goal of increasing the chances of success of your research projects. So we're trying to enable technologies to never miss a signal. I am in charge of helping to develop the software in the best possible way for the field of drug discovery. So Subir mentioned chemistry. We did make a few changes in the way that we approach the different areas of research at Revity Signals, and you can see the drug discovery box is a bit bigger because we are taking the approach really of combining chemistry and biology into drug discovery and taking a more holistic approach and developing solutions that take a holistic approach to treating the business problems of drug discovery and pharmaceutical research. In addition to other fields, of course, industrial chemistry, anything that is non-drug discovery is what we term either industrial chemistry, food flavors and fragrances, material sciences, and we have, of course, our suite of products, suite of software addresses those markets as well, in addition to clinical analytics. So, brief overview, Signals Research Suite, we have software solutions that address what we call the make, test, decide cycle of research. You make things, you test things, you decide which things you make next, and it's a cycle, starts over until you have optimized the right molecule, the right formulation. All of that being powered, of course, by industry-leading solutions, ChemDraw and Spotfire. So take, for example, you make a molecule that you are then going to test. You're going to test it in many different ways by many different people in many different labs on many different instruments, and all this data is diverse, sparse, and not normalized. So applications like, for example, Vitro Vivo, part of the Signals Research Suite, allow normalization and processing at scale. And once all those samples have been tested, then data analysis with proper visual analytics can then lead you to accurately choose the right molecule, the right formulation, the right biological to move on to the next iteration. So then once you have that, you make again, then you test again, and then until it's time to decide one last time whether that's a molecule, whether that's a biologic, whether that is a formulation. This is really the core approach that we have really to develop software solutions that address all the business problems that are associated with this workflow. Okay, and with that, across all of that, ensuring proper capture and proper processing of the data is critical. So just to take an example, this is AI generated image, of course. If you have a digital transformation strategy that involves moving from paper notebooks to a computer without a proper data strategy, without a vision, or without the right tools, of course, the problems with papers that you have today are just going to be translated into your computer, like the image shows. So couple of things that I would like to discuss and highlight for today's first presentation in terms of chemistry data, biology data, textual data, and instrument data. Those are all the areas of data capture, data processing that we have made, I believe, significant advancements in the recent years, and we're hard at work trying to keep improving that. So first, I'm starting with that because it's a topic that's always close to me. ChemDraw and making chemistry accessible. I don't know how many of you have ever seen this, but in PowerPoint, provided you have Microsoft 365, you have access to what is written on the screen, 3D models. And I once played with that as I was a product manager for ChemDraw and found that this was really cool. What is this? It turns out this molecule that you have seen rotating on the screen is a 3MF object. The 3MF is an open source 3D printing format that you can see the various companies that are part of this consortium. One of them is Microsoft. So Microsoft PowerPoint, Microsoft Word natively support the 3MF format. We took advantage of that, as part of making data accessible, making the communication of chemistry accessible, and we turned that into a feature in ChemDraw. So in the top left, this is a 2D molecule of ChemDraw, which since a couple of years you can now get a 3D energy minimized model, and then really from one click you can go from this molecule into either of those directly into PowerPoint. So you click a button in ChemDraw and you do Control V in PowerPoint, and then you animate. Those objects are natively animatable within PowerPoint. And another example, obviously, the primary usage of this format is animating in PowerPoint, but it's also a 3D printing format. So this is just something that I wanted to to bring to everyone's attention, because it's a very neat feature of ChemDraw. May not have been properly advertised, but here we are today. In addition, from the data capture and data handling point of view, we are moving in the direction of a modality-based drug discovery approach, right? So if you don't know what a modality is, I have asked the world's expert on everything for you. A modality is a type or category of therapeutic agent that is used to treat diseases. It's a term that you may already be using yourself, but you may see more and more in the literature. There are two types of modalities, what are called the traditional and conventional modalities. So the small molecule therapeutics, monoclonal antibodies for biological therapeutics, and the new ones that are emerging, and there are plenty of those. But if you think about PROTACs, if you think about antibody-drug conjugates, gene therapy, mRNA vaccines, and CRISPR, and stem cell, and oncolytic virus. So those are all the new modalities that are emerging and that are becoming part of the arsenal to treat diseases that were previously thought to be incurable. Right, it's pretty quite amazing what we can do. And so this is really something that addresses that sense both on the chemistry and biology side of data. Those new therapeutic modalities, you can see, the increase in size as you go from nucleic acids to peptides to enzymes to antibodies to cells to stem cells. And as they grow in size, they're harder to represent, they're harder to make, and they are harder to deliver. And of course, that makes the capture of this data harder and harder. So for this, we have really improved capabilities to capture and to visualize the natural and unnatural biopolymers with the Helm representation. The Helm representation in ChemDraw is something that we have introduced quite a while ago at version 2017, but we have recently, given the uptake of of new modalities and the request from our customers to better address data capture of those complex entities, we have gone from this to the possibility to visualize natural and unnatural biopolymers in a much more visual way. So that is really when it comes to data. Of course, there is the data management that you think of in terms of databases and connections, but in a very down-to-earth fashion, it's also extremely important how you interact, how you visualize data yourself as a human. We are all visual animals, and we try very hard to make complex data as easily understandable as possible. This is what we have done with the Helm capabilities in ChemDraw. And because of the way we build our software applications, if it's available in ChemDraw, it can be ported readily into other applications where you can use the web version of ChemDraw, such as Signals Notebook. So we have also made this Helm editor available within Signals Notebook with all the capabilities that you find in the desktop version of ChemDraw. And in addition to that, because we understand the needs from our customers, it's nice to have this tool, but the way that you are going to use those libraries of monomers is really in a centralized way. So this is proper centralized data management in that end users, so chemists at the bench or biologists or biochemists, should not be able to define any new biopolymer and then give them a name. Otherwise, chaos ensues. So rather, within a company, you should have a uniform, homogeneous nomenclature where a certain name is equivalent to a certain amino acid, for example. And this is where the centralized monomer libraries concept within Signals comes into play and allows a curator. You can see it really like a registration process and registration sort of curation process where one person or several persons are going to be in charge of validating and making proper definitions of monomers available to all the end users in Signals Notebook. Okay. Also, that was biopolymers with some chemistry to it, but we have also, from the purely biological data standpoint, made some strides in handling biological sequences. So Signals Notebook, for example, gives you the ability to search by DNA or protein sequences across the entire platform and match, the different traditional options for searching substring exact and nucleotide blast as well as protein blasts. In addition to the ability to handle RNA in experiments. So we are starting really from the point of view of nucleic acids and as well as proteins and peptides, and our goal is to continue addressing the proper data capture of larger and larger entities. This is all the sort of files, the sort of biological data that you can get, that you can see and play with and work with within Signals Notebook. Okay, then of course, so that was, excuse me, that was chemistry, biology data. Then we have handling paper structured data, everyone's favorite, and what is what is a worksheet. So there's a lot of information is captured in this way today, and a worksheet is really, as you all know, it's really controlled set of steps with checks that is traditionally done on paper. So it's semi-structured information that you entered. It's structured, but some fields are unstructured. So it's complicated to capture electronically in a structured manner. And it's most of the time, really a recipe or a checklist that you have to follow. It's often created as a PDF form, and it's often taken as a PDF scanned and then dragged and dropped into Signals Notebook, or sometimes created just directly within our electronic lab notebook, Signals notebook, as a text element, which doesn't really help in the grand scheme of taking advantage of your data at a later stage for data analysis or for later machine learning, for example. So this is why one of the recent, from last year, recent additions to Signals Notebook are worksheets that allow mixtures of input field and static text with possibility to lock, unlock fields, prevention of electronically signing a document if it's incomplete, having timestamps. It's about making it easier, making it as easy as possible to structure the data in traditionally unstructured data. And like, for example, excuse me, for example, entering a timestamp. I mean, this is really very, very simple, but as you are running your experiment, clicking on a button to insert a timestamp is something that is small but helpful. In addition to worksheets on the textual data management approach, we have made some great progress on enabling formulation entry, and of course without Excel. That is to say, giving the ability to define components of formulations and capture high-level metadata of the formulation itself, with either manual entry possibilities as well as importing of specific data from internal or external sources, with some calculations available, and we are continuing to heavily work on that. And another thing, this I believe was recently released as a beta feature. So if you are on Signals Notebook, you may request that. The possibility to create a variations table from a given formulation to allow description of individual formulations and the same features as the formulation table, but allowing you to make variations of one given formulation in a much easier fashion. Okay, and so that was chemistry, biology, textual data, and I just want to do a little introduction, quick introduction to before introducing the last type of data that is of interest for us. I just want to run through a couple of things that are high level. I'll try to make this as entertaining as possible, but if you, depending on where you are in your digital transformation journey, the only way you will take advantage of all your data is if you have the proper foundation for that. What I mean by that is the architecture. The architecture is extremely important when you implement scientific software or any other type of software. So an on-premise server, this is an allegory, my allegory for maintaining an on-premise server. When you will be looking at making an upgrade, this is what things will look like, and it may be costly, right? So you have two different considerations. You have on-premise servers and you have cloud servers, but you have two flavors of cloud that exist still today. You have cloud hosted, that I like to call cloudish, and you have cloud native. What's cloud hosted? Cloud hosted, if you want an analogy, is a bit like taking an old version of PowerPoint, an old version of Excel, an old version of Word, putting that into Dropbox, and then calling it Office 365. That's what cloud hosted means. So cloud hosted, if you go cloud hosted, you're just going to have the exact same problem in a cloud environment. Cloud hosted cannot, by definition, by this definition, cannot and will not scale, right? So you will start getting stuck, and so you will start getting performance issues, performance bottlenecks early on as data accumulates. The solution for that, and it's the solution that is used by all the major, for example, social networks on the planet, is cloud native. So cloud native technologies, servers you have providers like Microsoft, you have providers like AWS, and the advantage is that to increase computing power, you just increase the dial in an unlimited manner. I mean, as unlimited as your cloud budget is per month, because there are cloud server costs associated with that. But conceptually, if you have a performance problem, you increase number of nodes, you increase server power, and then performance problem is solved, right? So that is one thing to consider. Obviously, all the solutions I have been presenting before, Signals is built on the cloud native architecture that allows scaling. Another very important point of cloud native, goes to the topic of instrument integration, right? So how do you integrate various systems? Let's say conceptually you have the green circle is an ELN, the yellow circle is a LIMS. Traditionally, this was with on-premise servers. This would be how you connect and how you integrate between systems. You have plugs, you have connectors, and then connecting an instrument is really looks like this, and that's just one instrument. So you can imagine how messy it gets and how complicated it is. I'm sure many of you in large companies have dealt with that problem of integrating instruments. And without getting anything too technical, but I think it's just a general culture if you are in scientific software or in software in general, or you are using software in a modern way, the one benefit that cloud native technology is going to bring to you is what's called a REST API. What's a REST API? It's an application programming interface. And what does the REST API do? It allows transfer of data, connection and transfer of data in a secure and nimble way between various systems to exchange information, right? So rather than having all those plugs with all those connectors that hang, this is the modern way of exchanging and integrating between applications. So provided that your one software application has an API, a REST API, the other software has a REST API, so they can integrate and exchange information in a seamless fashion, right? So another exemplification of this is really point-to-point connections between instruments and electronic systems, between software applications, have traditionally been very challenging to build and very hard to maintain. It works fine with one, it's okay with two, but as you start adding more and more, it just becomes it looks like a subway map or a bus routes, and is completely unmanageable. So to address this, in partnership with a company called Cytera, we have recently launched our partnership, and the Signals DLX, DLX for Digital Lab Exchange, and it's built on top of Cytera's technology, which is called Platform as a Service, Integrated Platform as a Service, that takes advantage of cloud native applications that can exchange data in an easy way. So it allows to connect all data sources and make data integration from any instrument, any PC-based instruments, any data source, and make that available to all the other applications, provided, as we talked before, that they have an API. And this is something that you are able to do across excuse me, across various departments for testing, and connecting, in our case, for example, Signals Notebook experiments, entry of data into Signals Notebook experiments directly from a balance or from a Chromeleon run or from an Empower run. So this is about really facilitating and making instrument integration with electronic systems the way that it should have been before. Right? So with that, I did my best to try to not get us started and being late. Just want to do a quick recap. Really, Signals and Signals Research Suite is all about scientific data capture through a collaborative work environment with self-service data processing, allowing you to make data-driven scientific decisions. What's important is really in one user interface that is easy to use, modern, and it can benefit from all the modern software technologies. I did not, on purpose, dive into any specific video or demonstration in this presentation because I wanted to give you a high-level overview, and in 30 minutes, it is a little bit complicated to delve directly into one very specific topic. But if you are interested, of course, if you go on our website, revitysignals.com, on any page, any products page, any software products page, you will find this button on about all the pages, explore our interactive demo. And it's not a video, it's going to be more a self-service. You can click through and sort of experience the experience for yourself just in the web, what using the software, what it does, and what using the software may feel like. So I do encourage you, if you are interested, to just visit our websites. We're trying to make it as easy as possible for you to get a sense of what you can achieve with Signals solutions. With that, I want to thank all of you for joining us today, for being here, and I am very much looking forward to the upcoming presentations. Thank you very much.
Thank you very much, Pierre, for this informative insights. Really appreciate your time and for sharing the information. Thank you. Now, I just wanted to request everyone, if anyone has any question, please feel free to go to the question section and ask the question. Our panelists will going to reply all the questions in the Q&A session. Okay. Moving ahead, now I would like to invite Ms. Rupali uh, uh, for the session. And just for a brief introduction about Ms. Rupali. Ms. Rupali works as a head of uh, digital excellence in Cipla. She has an experienced pharmaceutical industry professional with more than 20 years of experience in product development, blended with the experience of digital over 10 years. She is expert in core product development, digital transformation, process reengineering, and regulatory compliance. Skilled in crafting the digital strategies with business priorities in various areas like development labs, regulatory affairs, and clinical management to enhance the productivity, foster collaborations, and improve time to market. She has implemented cutting-edge digital technologies such as AI, ML, BI, chatbot, and IoTs to drive successful enterprise digital initiatives like electronic lab notebook, SAP processes, document management system, modeling, and simulations for innovation regulatory information management system at Cipla. She has a strong background in computer system validation, providing a compliant framework for research activities, excellent stakeholder management, and expert in leading change management initiative for more than 1,000 plus users, facilitating seamless implementation for digital initiative and improving the employee engagement. Please welcome Ms. Rupali from Cipla. Ms. Rupali, thank you very much, and over to you. Thank you.
So thank you, Sukhbir, for this very detailed introduction. So good afternoon, everybody. And and thank you also to Reviti for giving me opportunity to present my thoughts in this very prestigious forum. So without taking much time, I'll move to today's topic, which is very interesting topic, is optimizing compliance in scientific research through ELN, and what is the best practices and the strategies for doing the same. So I hope you able to see my screen.
Yes, ma'am. We can see your screen.
Yes.
Thank you. Uh, so I call this as a very interesting topic because what is expected as a compliance in the research is not known fullest, because there isn't any published guidance from any of the authorities in this regard, similar to what we have for CGMP or GLP areas. So that is how this is still a very subjective kind of a topic, and uh, give me a moment, just some problem with the settings. Okay, so similar to CGMP and GLP, there isn't any clear guidance which is available for what is expected out of research labs in terms of compliance. And so every organization is actually developing their own framework for addressing the compliance related strategies for their individual organizations. And a lot of time these strategies are dependent on what are the current trends that are there in the industry about or what are the FDA or other regulatory health authorities views around this compliance. So moving to the next, just give me a moment, I'm not able to scroll through the screen. Second. So today we're going to cover that's how what is the trend of compliance in the area of research data, and how we can accomplish this compliance through lab digitalization, and specifically through implementation of electronic notebook. So we all are aware that the Indian pharmas are largely operating in the generic space, and in today's world there is no generic which has a simple kind of a product development, and every generic product is a complex product development. And because of this complexity, a lot of complex data is getting generated in the research labs of many pharma companies. A lot of complex data, such as the characterization data, and these data are largely correlated with the clinical data, and that is how it's becoming very critical data. And that is how FDA has started focusing on the studies which are related to creating these characterization data for product development of complex generics. And so Largely FDA has started auditing those products and the characterization data which are generated in the research lab, and that is how we are seeing a trend of FDA auditing research labs. And we see that the focus of the audit is not only understanding how the study has been designed, or how it is executed, or what is the conclusion of this study, but the more focus is given on to the data integrity aspect of the data and the associated lab processes. And that is how the compliance at the research lab, the expectations of health authorities at the compliance of research lab has an increasing standards that we see. And also that is how there is an establishment of development quality function that we see in every organization nowadays, which who are also constantly monitoring and supervising the data which is getting generated in the research labs. So how do we meet this increased demand of health authorities? So one is, can we elevate our current paper-based systems to address these increasing demands of health authority? The answer could be yes, but we all know that there are certain inherent constraints that we see with the paper-based system, and a lot of time we see there are constraints related to the reliability of the data. The data could be misplaced, or there are multiple documentation that needs to be generated for a single process. There is a lot of review mechanism that needs to be established in terms of making this data very compliant and compliant as against the ALCOA principles, and ensuring the complete data integrity is being maintained there. So that is how maintaining the paper-based system becomes a little bit inefficient when it comes to the managing all the principles of data integrity. But at the same time, the digital systems, when designed appropriately and adequately, have a lot of elements which can provide inbuilt compliance to all the data and the processes which are generated within the digital systems. And as per my perspective, electronic lab notebook is one of the centralized application which has a lot of processes, lab processes, covering a lot of lab processes within it, very key processes like all your experiment recording, as well as your instrument management, as well as connecting with the databases. So so that is how it brings a lot of compliance. So that is how once ELN is implemented in labs, it covers a lot of key processes within the labs, and all the data generated within the labs becomes very compliant. Also, through a lot of processes and workflows can be designed within the ELN, and that is how it can bring very real-time visibility to the processes, and which helps in improving the productivity as well. And also, as Pierre has mentioned, the data which is generated through ELN can be used for analytics, which can create robust product development. So what can be in scope of ELN? So as I said, almost all of your key lab processes can be embedded in the ELN. like recording of all the experimental data, including all your characterization data, including your stability data. Also, the instrument and the material management can be done through ELN. All of your important chromatography systems and key instrument and databases can be integrated seamlessly with ELN. Also, certain quality management workflows can be built within ELN. So that is how we see a lot of your key data can come within one roof of ELN, and how we can ensure the compliance of this data, and how we can ensure that the data meets all the ALCOA principles that we can see in the next slide. So this table depicts how a thoughtful and thoughtful design of ALN application can help you meeting your compliance needs. So the first column describes the principle of ALCOAs. What does it mean, and how inbuilt controls in ELN can help you achieve this. Though these are not the only controls that can be built depending upon the type of your records and the purpose for which these records can be used. There are multiple types of controls you can build within the system. Also, why I said it has to be very thoughtful design of a ELN system, because unlike other quality systems, this is basically used by research scientists. The system is used by research scientists, and that is how, while building the system, it's utmost important that we have a good combination of a flexibility within the system, as well as where needed we should embed the appropriate control so that the data and all the records which are generated within ELN meet the ALCOA compliance. So for example, I'm not going to read through everything, because these are just a kind of examples how you can achieve these principles. For example, this is attributable. That means who performs the activity and when the activity is performed. So ELN can be built with the control of date and timestamp. It also has a audit trail. Proper user management also can be defined, so we can understand who has performed which activity and when it was performed. Another is eligibility. Unlike paper systems, these are the electronic records, so there is no question of having a illegible kind of a record. Third is contemporaneous, which is the quick recording of any of the activity. So as every record has a date and timestamp and the audit trail, the scientists are bound to record the activity at the real time. The fourth is the originality of the record, which is the original copy or the certified true copy. So we can have certain ELN controls to ensure that this is a original copy. For example, e-signature, certain role-based privileges for editing the record, and so which can ensure that the records are original. The next is the accurate one, which is a error-free. So a lot of functionalities, such as making your experimental record as a templates, use of a lot of master data using the drop downs or auto text, can ensure that the data entry is accurate. Also, you can choose certain important fields as a mandatory fields where you can ensure that the scientists are filling this information, and this information is available for analytics. The next is the availability. So the retrievable, it has to be retrievable at any time for review. And this is the biggest kind of a issue that we observe with the paper-based system is the retrievability, and which is very well managed in any digital system. Also, you can retrieve the records through searches, various dashboards at any given time. Next is enduring, which means the records are available for predefined time. So you can, if you have a secure database structure, you have a defined archival policies, and the data is intact in the database. So which means you are also meeting this principle of enduring. The next is the data has to be very well complete, which means the data has to be in a complete state. So any electronic system, the data is not getting deleted. All the data, including repeat and reanalysis, are also available along with the experiment, so that there is no question about any hiding of a data or building any kind of transparency issues. The next is on the consistency. That means the record has to be reproducible. So as this is an electronic system, so all records are predefined along with their states and transition. So every such record will behave in the same manner for every user. So there is going to be quite a good reproducibility for every record. So this is so there is always question whether introducing such electronic system in research lab just to build the compliance, so whether it's going to affect my productivity because scientists will have to record so much of data, and whether it will cause any efficiency going down, any productivity going down for my scientists. Because as I said earlier, also the research labs are basically for scientists to record their observations, and that is how these systems are meant to be very flexible in terms of recording their observation. So while designing the system, there has to be fair balance around maintaining the flexibility as well as putting the controls over the critical aspects of a data. So the answer to this question is no. Actually, using these all principles of compliance, we produce a data which is a rich quality, which is of a rich quality, and we call it as a fair data, which is findable, attributable, interoperable, and reusable kind of a data. And this data becomes the basis of your analytics. Using this any analytics tool, scientific dashboards can be created for scientists to take data-driven decisions. It can give a lot of insights to scientists, and which helps to them for robust product development, and which ultimately results in a greater speed to market. So having the compliance in place for recording of data or for all of the lab processes doesn't reduce the productivity, but it actually helps to build the good data which can be utilized by the scientists to understand the product in a better way. So thank you for allowing me to present this, and I would be happy to answer any of your questions. And Apologies that I had a bad throat, so initial few minutes I couldn't be of very uh, fluent.
No worries, ma'am. We can understand on that. Thank you very much for being with us and giving informative insights on the ELN. Moving ahead, we would be moving with the third presenter for today, uh, Tanvira Paul, who is the field application scientist at Revity Signals, and she would be talking about very interesting topic: unlocking efficiency, biosimilar research in Signals notebooks. Just to give you a small brief about the Tanvira, from a qualification, she holds a PhD in biochemistry from PIT Vellore, and at Revity Signals, she plays an active role in as a field application scientist in biology, and she is in charge for sharing her technical knowledge with the pre-sales presentations, showing how the good the company products are, and setting up and keeping them in a good shape. She also assists the sales staff in assessing the potential applications of company product to meet the customer needs and preparing a detailed product specification for the development and the implementation of the customer's applications and solutions. So over to you, Tanvira.
Hi, Sukhbir. Good afternoon,
Yeah,
everyone. I'm really sorry. There was some technical issue with the unmuting. So here I
no
am.
worries. Over to you, Tanvira, for your presentation. Thank you.
Thank you, Sukhbir. Is my screen visible before you leave and I sound okay?
Yes, your sound and your screen are absolutely perfect. Thank you.
Okay, I'll take it from here. So good afternoon and hello, everyone. Thank you, Sukhbir, for the introduction. And thank you, all of you, for taking out time and being here. I am really honored to be present in front of all of you and would like to take this opportunity to take all of you on a journey to exploring our one of the best in the class product, Signals Notebook. Now, our Signals Notebook is a part of a more unified, larger and integrated system called the Signals Research Suite. Pierre had already given an overview of our product, but I would now take all of you to witness how this works within the Signals Notebook. Signals Notebook comes into play in the make stage of the make test decide life cycle. Signals Notebook is increasingly and has been widespreadly being used by chemists for different sectors, industrial sectors of drug developments. But now we have slowly crossed over to its applications in biology as well. So in this particular talk today, I will be showcasing as to how the S&B can respond to the various experimental needs of the research professionals or the scientists. Our platform, in a brief overview, gives you an entire control over different data sets, different techniques. With different integration techniques, you now have everything in the single platform. Signals Notebook is also an amazing collaborative platform because it facilitates both internal and external collaborations, maybe within the organizations or outside of the organization, irrespective of what the locations are. Before talking more in details about the Signals Notebook, let us first see a little glimpse about our Signals Research Suite. Our Signals Research Suite is a single SaaS package, and it makes the entire research life cycle much more easier and much more time efficient, and also makes you more productive. And we will slowly explore and see how we can unlock all the efficiencies that are stored in our Signals Notebook as capabilities. Now, our Signals Research Suite comprises of the Signals Notebook, as you have said, and we have other two softwares called the Signals Victrovivo and Signals Inventa. while Signal Notebook helps you in capturing and organizing your ideas, your data that is required for creating your experimental workflow. Signals Vitro Vivo comes into play in the test stage, where you need to test all your raw data from the experimental parameters. All of these can be calculated, analyzed, visualized, and can be published into the Signals data factory. And Signals Inventor is our third product within the Signals Research Suite that helps you to compare all your overall assay results, and thus helps the research professionals to make data-driven decisions or informed decisions regarding the drug discovery process. Now, in this session, I'll be focusing mainly on a biosimilar workflow. In this case, I have considered a biosimilar insulin production. Now, we know that biosimilars are drugs which are similar but not identical to the original drugs. The kind of compound integrations or the kind of bonds they have between them makes them even more complex. And Pierre had already mentioned how as other molecules get bigger and it becomes harder to represent or make or to deliver. So Signals Notebook, the Signals Research Suite as a whole gives you the capability to deal with this more complex molecules and carry forward with the experimental workflow. It helps you to digitize your entire experimental data. You can compare your old data, reuse the data, thereby automating the entire process. And since it's a single and common platform, your results is guaranteed to have a consistency throughout the experimental cycle. Now, as I told that I'll be considering an example of the biosimilar insulin production. Insulin is a smaller molecule, but it is still bigger as compared to a chemical molecule in this case. So these are the steps that are usually involved, a generalized overview of how a biosimilar insulin production looks like. There are different stages in the experimentations. Initially, a scientist starts by establishing or optimizing their cell line development, which can be carried out in organisms like E. coli or yeast. In this case, we are considering bacterial organism for the cell line development, later to which the scientist focuses on isolating this cell lines that is capable of consistently producing the insulins, and go for the bioprocessing or the upstream processing, where fermentation is being carried out and the bacterial organisms are being cultured for larger production of the insulin. And before going to the downstream processing, there are initial post-processings, after which we proceed to our downstream processings before we eventually extract or harvest the insulin and formulate it for to be marketed in the commercial market. So now we will see how the Signals Notebook comes into use or comes into play with its features and capabilities that enables and makes this biosimilar insulin production quite easier, and quite easier with the time management and the productivity, and not the process, because it is still complicated. But we will see how it can be eased down so that it becomes more profitable and time saving. So the scientist begins by logging onto the Signals Notebook, which is end user specific. And since Signals Notebook is uses cloud native technology, so you have user specific web address, and to access the Signals Notebook, it requires it is a zero installation software, which makes it quite efficient to be used and has a better and a faster performance as compared to the on-premises softwares. So let us now move on to the Signals Notebook. Once the scientist, which is in this case is me, have logged on to the Signals Notebook, we will start with capturing the cell line development workflow. So in the experiments, we can see the following features, and we will have a glimpse of how this can be done. In this particular case, you get to see the experimental workflow that was set up by the scientist. In this case, she goes ahead and she sets forwards functionable aim that is required, and to support and see the overall picture of the experimentation. She then finds out a certain supportive materials and paper which she found was extremely helpful for carrying forward the experiment. So she can quickly go ahead and drag and drop it over there, or you can also upload the file from over there. After moving on with the initial setup, she goes ahead and checks with the experimental requirements in the experiment. As you can see, he goes ahead and chooses. We have an inbuilt materials library, which is the house for all of your experimental needs and wants. Not only you can choose your equipment from a list of hundreds and hundreds of equipments, but you also get to pull on the data that are associated with this equipments, like their calibration dates or their details or their manufacturers. This is not limited to just equipments, and you can add a lot of other components like consumables or your cell lines or your biological samples, all of it. You already got a little glimpse of how other biological samples are maintained within the libraries. The biological sequences are visible in the dynamic mode within the materials library and also within your experiment. We will come to that later in the later part, and we'll see how they appear. So here you can see that the plasmids biological sequences are visible to everyone, and it allows you to copy. We have a seamless integration of SnapGene, which allows you to visualize all these sequences and lets you also copy the subsequences in the reverse format, in their original format, or they have got, and also gives us some features of downloading these sequences in certain formats, in certain specific formats. So after these plasmids, or the biological sequence, in this case plasmid, the biological sequences can be the DNAs, RNAs, or the proteins. They also come up with all the essential functionalities or the features like the melting points or the restriction signs, all of them inbuilt within the materials library for that particular component. So once we are done with the experiment, adding our biological sequences and equipments, we start with the experimental flow, and eventually after culturing, after carrying out the transformation of the bacterial organism, we do the blue-white colony screenings, and we can even register those colonies and those plates within the materials library. We not only get to register the plates, but we also get to register our samples, which in this case are the white transformed colonies, which can be linked with their experimentations, the date they were created, and all these details were brought into one place in the signals notebook. Here, the samples were created from the picked up colonies, which were then submitted into the materials library, and then further, without going into the hassle of contacting your fellow researcher or colleague for carrying out the next steps or the analysis of the sample, we can directly create tasks for each of these samples and assign a particular person. As you can see over here, you can assign a particular your colleague or your subordinate or somebody with these tasks, which can be seamlessly performed without even you migrating to a messenger or a mailing system. So, and as you can see over here, these tasks are pretty well defined, and there is no chance of any confusion or an error or a spelling mistake, in when we usually write and do the things manually. So registering your samples and creating the tasks are really very easy and very simple, and saves a lot of your time within the signals notebook. So once I have assigned my task, the colonies are required to be tested. They are to be used for fermentation in bioreactors. So for fermentation process to carry on at different stages of the fermentation process, where we are upscaling the production of the insulin from these microorganisms or from the bacteria, we need to carry out samplings and also figure out their quality by performing various experiments. So from in the fermentation protocol itself, you can carry out your experiments, the plate-based assays. We all know, like for most of the scientists, in fact for every one of them, maintaining the data in the plate physically is and manually is a very tedious, and it's a humongous job. So Signals Notebook has now come up, has now this improved feature, which is a plate-based features where we are submitting our samples for further analysis. And you can see that we can map out our plates not only by by the kind of samples that are being used, but if you can go ahead and see, we can adjust and write their concentrations, link the material IDs or the samples that will be tested, causing no anomaly, because all of these are present in all different forms. I mean, like all different pages where you can see that the concentrations, the amount, all of this can be documented directly, and this helps you to seamlessly uh, document all organize your data within the plates. And from these plates, our the other two products, which are the Signals Vitrovivo and Signal Inventa, seamlessly helps you to visualize this data directly, visualize and analyze the data directly from the Signals notebook, and as such helps you process all your raw data into actionable results, thus helping you to make more informed decisions. So in summary, it can be said that the Signals notebook is a platform that helps you to organize and capture all your unorganized data or scattered data, so you do not miss out on anything or make any kind of mistakes while you're creating your workflow. It helps to search any sorts of datas or images or plates or results, samples, everything within the system. It can be done with a click. And we all know scientists really do not like to waste time in searching for their needs or requirements. So it makes searching your data quite easy. It helps in collaboration, like you can see in the experimental workflow that I showcased. The one scientist did not require to come out of the system or the platform to go ahead and collaborate with the other scientists for the entire experimental workflow to be concluded. And lastly, when your experimental workflow is seamlessly being created and you have all your data analysis softwares and all your visualization tools in the single platform, it's nothing but it makes all your process extremely time efficient, making you really more productive, and thus gives all the industry or the professionals more time to invest into more productive research, thereby allowing them to put more useful products into the market. So with this, I am ending this talk or on our Signals notebook. This is our hyper trial system where people who want to have like a view or visualization about how Signals notebook functions or works, you have got minimum configurations that are already been set within the system. So if you would like to go through it, you can try our hyper trial system, and within registering, within 3 minutes, the system gets activated and you get to have your hands on. So thank you, everyone. If you have any questions, you can submit them, and I'll try to answer the best of my knowledge at the end of the webinar. Thank you so much, and over to you, Sukhbir.
Yeah, thank you, Tanvira, for this insightful session and presentation. I would like to re-emphasize once again, if anyone has any questions, please feel free to ask. All panelists will going to reply to your questions in the Q&A session. Now, moving to our fourth presenter, Mr. Rangarao. So Rangarao is a chemist with more than 25 years of experience in drug discovery chemistry, both in the infectious and the tropical disease. He is currently working in the drug discovery chemistry at Bugworks Research, and he's involved in managing the contract research organization's chemist. Previously, he has worked in Novartis Institution of Tropical Diseases in Singapore, and AstraZeneca R&D India, involved in the drug discovery programs targeting the tuberculosis, African sleeping sickness, and dengue fever. So I would like to invite Mr. Ranga Rao for his presentation. Sir, over to you. Thank you.
Thank you, Sukhbir. Good afternoon, all. What I will do is I will switch off my video because looks network is weak. Can you hear me all?
Yes, sir, we can hear you perfectly. Thank you.
What about my slide?
Yes, we can see your screen also, sir. Thank you.
Okay, thank you. Hey, good afternoon, all. My name is Ranga Rao. I'm from Bugworks. I'm a chemist. I am responsible for all the chemistry deliverables for all the projects in Bugworks. Let me quickly take you to my topic, transferring startup dynamics in pharmaceutical, digitalization for accelerated innovation. So how digitalization will help in accelerating the discovery company like us, like Bugworks. So just to give a little background about Bugworks, Bugworks is a small biotech company based in Bangalore. It started 10 years back with a goal to discover or hunt a new drug for antimicrobial resistance. So I'm happy to share that we already have discovered a novel broad spectrum antibacterial agent with a lead candidate in clinical trials in progress. And in 2020, we expanded our research area to oncology, and we have multiple projects in different stage of the discovery, treating both in immunotherapy and targeted therapy. The most advanced project is we have a novel anti-cancer agent with a lead candidate ready for IND submission. With these two unmet medical need areas, we are continue our drug discovery research until we succeed. We are funded global investors throughout globally, both from in Asia, Africa, Europe, and from US. We have a drug discovery platform built in-house, starting from target identification and validation to lead identification, lead optimization until pre-IND filing or a preclinical candidate. Just to emphasize on the Revity software, whatever my previous speakers updated, plays an important role in our journey, starting from scratch to until the compound goes to the market. So drug discovery is a multidisciplinary collaborative effort. We collaborated more than 30 to 40 collaborators or the partners across the globe. We can see Revity is also one of our collaborators. So I joined Bugworks in late 2020 when we expanded our drug discovery disease area to oncology. We have multiple projects. We do not have in-house wet chemistry lab. We outsource all the chemistry activities to 2 CROs. The major challenge we found, particularly I found, is monitoring these CROs remotely without communication is a major challenge, particularly to monitor productivity and transparency. The only way we can communicate is through project update meetings and emails. So since we are a small biotech company, we don't want to invest on IT person or IT support, and we don't want to have our own in-house server. So once we are looking for a tool or a platform with which is user friendly with a minimum training requirement, and also a tool that widely validated and globally accepted. And ours is a drug discovery company, so we need to make sure our IP is well protected and secured. And in case any kind of disaster take place, it should be managed and restored all the data without compromising on the integrity of the data. So with this, we introduced our Signals Notebook in October 2020. Before introducing this, earlier it is called Parkinson Signals Notebook. And before introducing this Signals Notebook, we explored most of the top marketed products in the market, particularly all electronic notebooks, and finally the PerkinElmer stand out to and fulfilled all the checklist we had at that time. The main feature we liked in Signals Notebook is the inbuilt camera. No camera, no chemistry. Where there is a chemistry, there is a camera. So, and one more thing we liked is all the features that have in camera is already inbuilt right at the Signals Notebook. So Signals Notebook is integrated with Microsoft software, so we can upload any kind of files, including images, by simply drag and drop interface. And this Signal Notebook is also will helps in sharing knowledge within the team and across CROs. Particularly if a chemist wanted to initiate a new experiment, first he can search, say the particular experiment is already performed, is available inside the Signals Notebook, and you no need to spend time on searching for the literature to perform a particular experiment. And also you no need to write everything. You just need to duplicate the experiment. You can modify what all the key features or the reactants, what are the reagents changed in that particular experiment. So with minimum time and minimum efforts you can spend, you can perform experiment compared to earlier using paper notebooks. And also the data generated are legible with a uniform scientific format. Particularly in chemistry, what we have done is we shared a standard experiment model to follow all the to all the chemists across our CROs, and so that the format will be uniform, and the data or the information entered in each section will be the same. This format will not be possible in a paper notebook. And we can customize the data format what you want in a Signals Notebook based on your project needs. Particularly, this will be very helpful for a biologist because they run multiple assays of different assays, and the format will not be the same compared to our chemist experimental format. One more thing we liked is the ChemACX is a chemical database inbuilt inside the Signals notebook. It's a collection of almost 20 million substances and 37 million products from 780 vendors worldwide. And it is updated every 3 months and provides an accurate supplier information, package size, purity, and price. This is very helpful for our CRO chemists, and amazing searchable options. Chemists love to search all the experiment by structure search, and you can search by dates, name, cache number, and also we monitor the progress or the productivity of our CROs by date wise. By selecting the CROs, we can monitor the productivity, and also the individual chemists productivity also we can monitor by search. And also we can access the data at any time instantly with full security. And one more important thing we liked in Signals notebook is the real-time stamping. Any kind of activity or the changes you made inside the Signals notebook, it automatically recorded in the background in real time. And this will help for the audit purpose. And we can also give access control on need-to-know basis. And we can give access within the team as a standard user or administrative control users. So there are different types of access controls available. You can choose whichever you want based on your need-to-know basis. And data management, we have not found any kind of issues from the last 3 years. So coming back to our next digitalization journey, we already implemented our chemical inventory database management, and we initiated the compound registration in the Signals notebook. In the future, we have planned to introduce a Vitro Vivo platform and also Inventa, where these tools help us whatever the data we generated from all our customers or partners and in-house will be managed and analyzed, and also Inventa to draw scientific decisions using Inventa. So with this, Signals notebook definitely helps each and every person, everyone in the team, from the end users until the clients. Definitely it enhance productivity and improved workflow. And one more important thing we liked is you can retain the ownership of the data, even the researchers are moved on. The major problem you are facing in CRO is the turnover of resources within the CROs. So even if the chemist or the researchers move on, we still own our own ownership of our data. With this, the take-home message is Surely, with by using effective collaboration tools and platforms, we can facilitate and accelerate innovation, and and also it helps in to take a better decision to increase the chances of success. With this, I would like to thank Riviti and also all the participants. If you have any questions, I would like to happy to take your questions. Thank you.
Thank you very much. Thank you very much, Mr. Rangarao, for the beautiful presentation. I really appreciate one of the line which you said in the presentation. Wherever there is a chemistry, there is a chem draw. I truly acknowledge this line. Thank you.
Yes.
Okay.
Thank you.
Moving ahead, we will have a small poll now, and may I request Arshad to bring that poll? Yep. May I request all the participants to have a quick poll participation? We will have a 60 seconds. I'll just read the question for everyone. Which Revati Signals workflow or features do you find most beneficial or would like to learn more about? Material libraries and inventory, sample and the task request management, ease of data capture and search, or all? Thank you.
Can you hear me, Sukhbir?
Yes, Manish, I can hear you.
Right. Okay. Okay. Thanks, Sukhbir. Thanks for the introduction, and thank you all for joining us today. And let's start. So, Sukhbir, hope you are able to see my full screen, and let's start. Okay.
Yes, Manish, I can see. Thank
All
you.
right. Okay. Okay. Thank you. So I'm going to talk about how we can help you with the formulations workflow. And to start with that, in our day-to-day life, just hold on this screen. In our day-to-day life, we are surrounded by a variety of formulation products such as pharmaceuticals, cosmetics, paints, agrochemicals, lubricants, and so on. And development of these formulations focus mainly on understanding how combination of different materials can provide desired performance for specific application. Certainly, the scientific complexity in performance optimization and reducing the cost and time to market are some of the key challenges in formulations industry. Of course, discussing formulation development in detail is not in the scope of this session, but I'll touch upon some of the key aspects of formulations development. So when we talk about any formulation, regardless of the type of the formulation, it involves some common phases, some of the common key aspects, which are like it starts right from project planning. It requires brainstorming on the scope, market intelligence, and feasibility. It requires critical assessment of the materials. Extensive documentation is required in compliance with the regulations. Huge amount of data is generated in experimental work, and all that information generated throughout this process needs to be carefully analyzed to make better and faster decisions. And if you look at this whole process to address these challenges, it requires enhanced collaboration, ease of data capturing, unified data storage, and how faster you can make a decision that determines how quickly you can reach to the market. So with these requirements, we're going to see how Signals Research Suite can help you with the data capture and collaborations using Signals Notebook, and help you in making better analysis, visualization, and data-driven decisions in a Signals environment. So as discussed in this process, it starts from project planning, where project manager would like to create a new project. A project manager can create a new project, generate a project code, capture related information, the scope of the project, and market analysis and background information he can capture. Then circulate this information to the key stakeholders. He can form his team. Different groups can be formed within the system. He can communicate with R&D heads, let's say analytical R&D head and QAs and formulation heads. They can form their team, assign roles and privileges to individual group leaders or team players. That's how they can communicate the project related information with them. Also, it requires a lot of documentation in form of several PDFs, Word documents, and PowerPoints regularly. Project update presentations, Gantt charts are required, and all this can be created within the system with integrated Office 365. So you can create your presentation straight away, update your minutes of meeting, email files, which you can circulate, and also you can communicate with the chat boxes. So that can help you in communicating project related information. Once the project is created, of course, it requires to check availability of materials, whether required materials are available, what is their cost, who are the vendors, or you need to go through the procurement process. And for that, within the Cygnus notebook, you can have the inventory capabilities, which makes searching all these chemicals much easier. You can search these chemicals by locations, or you can search this by locations or by containers. The locations can be organized in a hierarchical manner. For example, your business site, and then specific R&D, and then building, and the floor in that building, the refrigerator, in which shelf of the refrigerator or a drawer, and then so on. The sublocations, that's how you can organize all your materials. You can access all the related information quickly. Check if those materials are available within the lab, and what is their status, how much amount is left. If it is not present, you can also order new material. Even the scientists can initiate order. They can check the material information. You can create a material order by entering even a chemical structure or a gas number or even a vendor information. Dr. Rangarao mentioned about ChemSCX capabilities. So ChemSCX has more than 22 million compounds with all vendor information updated with their pack size and material information. So you can enter gas number or structure. It can perform structure substructure search, and that will help you in so that way you can initiate something went wrong, that's fine. So you can initiate a material request. You can check all the vendors, their information, fill in the order form, where the destination for which project you want to order, the size of the container, all these details you can enter, and then you can place order. Once the order is placed, you can even track those order status, whether it has been received, and once it is received, can get acknowledged and added to inventory. So even when you add to inventory after receiving, you check if that material is already present in the lab or it's a new material. So you go through the registration process where software can help you in assessing the duplication or creating lots and batch for that particular material. And then you can create a number of containers, generate barcodes, define the location or home location for those containers, calibration, or its expiry dates. All this information you can enter, and then finally have those containers in the desired location where you can use it. So even when you receive this material, it has the integrated PubChem integration, so you can update the material properties from PubChem, and you can have all this container information updated. You can so as it shows, you can have this max rate. You can look at the containers, you can check their properties, you can update their properties from PubChem, or even refer those properties from PubChem, and that's how it can also brings you the safety, hazard, precautionary statement information. So once we have a initial phase of brainstorming, you plan your project, and then afterwards you look at the availability of materials. If material is not available, you order, and materials brings lots of documentation, the specifications of these materials, then handling conditions, and then control procedures to test those materials. And throughout the formulations development, a lot of documentation is required that has to be in compliant manner. And typically, the paper-based system managing these documents is a big task. And where in this system you can create all those kind of documents with built-in Office 365 capabilities. You can create your Word documents, or you can upload PDF documents. You can cross-reference those documents, whether it's SOP specifications, protocols. You can cross-reference those protocols within your experiment. Having preset filters will ensure that you will always have access to the latest version and the approved version, so it can take care of the access controls. It can help you with the archival. Moreover, it can also help you in managing the workflows, the access controls to the SOPs, also like who can review and approve. So you can have the QA approval or individual department head approval, so that those approvals can be defined within the system. So once you have project is planned, materials are organized, and then documentation is in place. Of course, this doesn't go in a linear fashion. It goes in cycles. Experiment execution is one of the major tasks, major resource-intensive tasks, where a lot of different departments and analytical or formulation techniques and scientists involved, and they perform a variety of experiments. And that all that information, what bench scientists expect is ease of data capture and how efficiently they can collaborate, which can make their life easier and capture all the data in accurate and contextual manner. So for that, in previous talks, you have heard about Cygnus Notebook. It's a cloud native ELN. What that means, you need just a web accessible device where you can put this URL and start using this system. And the moment you enter, you can have your dashboard. So even before you start your work, you can look at all the activities going on. Then you can plan your work, and you start working on the system, and you create experiment. Let's switch the gears, and since we have some time, I want to take you quickly to my live system, because I believe you have seen a lot of screenshots. So I want to give you a quick glimpse into the live system, and then I'll come back to wrap up to my PowerPoint presentation. Okay. So I have already opened my Cygnus notebook, which you can see it here. And I'm going to show you one example of a formulations experiment. And what you are seeing here is this is the first glimpse of my Cygnus notebook. And once documentation in place, materials are available, formulation scientist would start his experiment. For that, he can create a new experiment. So it is important when you create experiment, it is it captures all required information and the metadata, and it interlinks. So it can be templatized. Formulation scientists can select template and create new experiments. So this is the one experiment has been created where all the information such as which department and type of experiment, all this information formulation scientist can capture, record his aim and background with all word formatting capabilities and auto text features. You can easily and efficiently capture all aim, background and procedures, this information. Before you start your experiment, you cross-reference which SOPs or equipments or reagents which you have used during the experiments. So here you can see the SOPs. As discussed, the SOPs can be created within the system and you can cross-reference. So within a few clicks, you can search for SOP, click here, and it will take you to all available SOPs, which are always preset filters, shows it is approved, and then you can select that SOP and you can bring it here. Similarly, Tanvira explained about different kind of materials libraries. All the materials you make in-house or even order commercial materials which you order, you can have different material libraries. You can organize all these materials in different libraries so that you can use them and cross-reference in the experiments. In this case, variety of equipments which you use to perform the experiment. In this case, you can go to query your different libraries, or if it is a reagent I have selected. So this is the one reagent I have selected, and I can quickly select and add these reagents to my reagents library, and that's how you can quickly search query materials libraries for SOPs, specifications, equipments, or reagents. As you can see here, I brought this one reagent. It brings this information. Also with the PubChem update, it brings this hazard information, safety precautions, this information along with these reagents. After reagents, the main part of the formulation is capturing formulation composition, your recipes. So you can capture which is your reference listed drug. So in this case, the reference listed drug again can have its separate materials library from the active pharmaceutical chemical, which can be brought in here, and then you can start editing a material composition. So what it helps you in capturing all the composition. For example, this is a 500 mg tablet preparation, unit mass plan, batch mass plan, and then different kind of materials. The same way you can query these materials, and you can define their roles. What is the role of that particular material? You can have drop down menu. The materials you can bring and see their information. In addition, it requires the calculation of the delta calculation between the planned and actual amount. So it has the built-in calculation capabilities. So you can configure calculation formulas, and you can calculate these values. So now this is the one single formulation, but often in formulation optimization, you need to work with a series of trials. You have to design multiple trials with the changing compositions. So of course you can create multiple tables for that, but Pierre just mentioned in his showed one feature in his presentation, which is still in beta, and I hope you will have access in Cygnus notebook very soon. But I just want to give you a quick glimpse into that. So particularly when you want to capture formulation variations, different formulation recipes, and want to review all those formulations at the same time. So the same information can capture like this. You can have all your common components which are required, and in screening experiment you can have the varying ratios of these materials, or even some different grades of the materials. So you can capture, again, same idea, you can capture these materials, and then afterwards you can capture variants. So for the same recipe, it can have different variants, variant 1 or variant 2, and that's how you can create multiple variants without a necessity to re-enter all this information. So if I click add variant, quickly it can add one more table which will bring all this information, and I just have to make changes in my material. So how much quantity I actually need. For in this case, this is 725 and this is the batch size is 700. You can capture all this information. Now having this information, you can download this to CSV, or you can copy this information, reuse this information. Apart from capturing formulation composition, you also need to capture, formulation scientists need to capture a variety of unit operations, and for that there can be a protocols. So you can have the protocol with a series of steps, right from the dispensing, sifting, blending, granulation, and up to the drying. So date and time stamp, and whether it has been executed or not, that check mark it can have. So variety of steps can be organized as a protocol, and then afterwards you can also attach images, do the annotations, and then this is the one, like first step is a sifting or granulation. If you have a DOE based granulation studies, all this kind of information you can capture, you can create table and have it in a systematic manner. Advantage is that having this templatized process, every time scientists will capture information in a contextual and similar fashion, so that it will help you extracting information easily. It will help in making better sense of all the information that you capture. So after you have captured all the unit operations, and you can attach different kind of images, your visual observations for this. Formulation scientists, of course, they need to they create analysis samples, or also need to initiate the stability studies. They can create stability pool schedules, and then the different kind of protocols can be created, and within the system they can manage different kind of analysis for this sample. So even before they create a sample, so once a formulation has been created, formulation scientists can create a variety of samples, right, starting from say blending process to coating, lubrication, and tableting. At each of the step they can take multiple samples, and samples will have unique identification. They can have the date and time stamp when it has been created, and then they can create samples task for this sample. Different kind of analytical test requests they can create for this sample, and those can be like characterization, stability, or different kind of test forms can be created, and those tasks can be submitted to analytical chemists so that they can perform analysis. So within the system you can have the we call it R&D LIMS workflow, where cross-department communication can happen. Formulation scientists submit samples, and analytical scientists can perform that analysis. So as you can see, this particular sample was submitted, and analytical scientist has performed. So right away from here I can go and check that analytical experiment, what has been performed, and also I can see all the data which analytical chemist has sent back for this particular sample. And similarly, you can have the stability studies and those CSV files. All this information you can organize in one place, and that's how you can have end-to-end workflow for your formulation. At the end, so of course, once the experiment is completed, you need to go through the sign and close process. As a compliance requirement, you have to go through the reviews. You can have multiple reviewers, multiple stages of the review, and then system keeps track of all the activities, keeps the audit trail, and finally you would like to take a report out of this sample. You can click print to report, and you can select what you would like to see in this particular report. So with one click you can bring all this information. It will have the report, and this report is not only the pictures, but what you can see in this report, you can even see all the attachments which you have in a native format. So whether in this experiment you have attached different PDFs and Excels, and all this information you can access here within the report. Okay, so with this I'll go back to my PowerPoint presentation, and just quickly hold on. So this was just to give you a quick glimpse into the system, and with that we have just discussed sorry, I'll just skip some of these slides. So we have just discussed this workflow, and then how you can create a report which can capture entire process. once you have performed all this experimental work, project planning, materials information, all this information gets accumulated in one system, and then you have to make sense out of it. You have to analyze this information to make better decisions. One of the options in Signals Notebook, of course, you see all this information as a dashboard. Quickly you can access, managers can access different information, for example, inventory information, equipment calibrations, or there can be different review dashboards. So the dashboards can help you in accessing all this information quickly. It provides you a variety of search filters, which helps you in easily accessing all the information. With a few clicks, you can bring all this information. Imagine the paper-based system where you need some information and it can take your days. So now, apart from this, on top of this, all the information which gets accumulated inside the system is index, which can be, you can perform global search, and then all this information can be fetched in a data analytics where Inventor comes in a picture. So you can bring all this information and you perform advanced analytics on that. So this is one example where it's like an initial feasibility study where you multiple formulation trials have been taken, 8 trials have been taken, and this is the one, the reference batch, and have to check, scientists would like to see which one is closely matching to the reference, and that's how you can compare different batches, their compositions, and some of test performances, so like physical test, free abilities, bulk density, and the release profiles. All these tests determines success of formulation, and you can compare profile of each of this batch here, and you can identify which is a closely matching batch, which batch you should pursue further for scale up activities. And of course, the reducing cost and timelines is one of the objective. So you can look at the cost estimates for individual batches or how it compares with your reference batch. You can look at the science as well as the economics, and then you can look at the analyze all your stability data, look at the out of the trained analysis, and you can do all statistical analysis, compare a variety, like dissolution is one of the major test in a formulation. You can compare dissolution profiles, batch to batch comparison. You can do this advanced analytics. With this, I would like to summarize. We have seen these things. So throughout this process, right from project planning to analysis and control, we help you with unified data environment. It can help you in enhancing the collaboration efficiently. You can capture your data. All the data you capture will be contextual, accurate, reliable. It will fulfill your Alka principles, what we call, like it has to be attributable, legible, and contextual. Those principles it can follow. You can meet the compliance requirements. You communicate seamlessly with your colleagues, different departments, using chat boxes, email notifications, or a variety of tools that system provides. More importantly, it provides enhanced user experience. It provides intuitive, incredible, fast Google-like search experience. You can access all this information. You don't need any external IT help to access and analyze your data. And with Inventor, you can make further analysis, advanced analysis, make better decision, visualize your data, and that can help you in making better products faster, more efficiently. That can help you in accelerating your formulation development process. That's all. Thank you.
Thank you very much, Manish, for this session. It's always great to see a live platform. Appreciate your time and your for giving a presentation. So moving ahead, we will have a last poll before we can move on to the Q&A session. Harsha, can we have a last poll for our audience? Okay, and I will read this question for the audience. Which of the following statements about the enterprise laboratory information system applies to you? Currently do not have any, but are exploring the market. I have previously used it but stopped. I have one, but looking for a better alternative. I would request all the audience to answer this poll. Arsha, can we have? Okay, so we have 67% says currently do not have it, but are exploring the market. So definitely we will be in touch with you to see if we can, if Revity Solutions can help you with the available options. I have one, but are looking for an better alternative. We will definitely connect with you also to see how we can help you to ensure that the proper ROI of the existing solutions can be driven out. Thank you very much for participating in this poll. Now we will take up the Q&A part. We have got a tremendous response from everyone in the Q&A part. And may I have the panelist also on the screen? Yeah, and we basically asked a lot of questions. Uh, we basically got a lot of questions from the audience before the session also. So what I will try to do is I will try to ask the questions to the panelist. However, if I miss any of the question, since we are recording this session, we will reach out to you uh, for the specific question which we are unable to take up in this Q&A part. So to start with, I would start with Pia first. Pia, you have given a very good presentation in the beginning itself, where you have talked about the transformational role of the data management in making informed decisions. However, we have also seen that people or the users who are basically using the cloud based solutions, especially the Signal notebook which we have, they have a concerns over the security part. And one of the question is covering this part also. It will be great if you can help us in sharing more insights on the security part, especially on the compliance requirement and the data, how data is stored on the cloud.
Sure. I'm going to answer in a very succinct way, which would be if you have security concerns, the quick answer is we are perfectly SOC 2 compliant when it comes to the offering, the cloud offerings that we have. So there are different flavors, of course, of we have different flavors of Signals Notebook when it comes to cloud, different offerings that I am not a full expert on, but we have Signals Notebook standard, E3, and private, right? And private is really Signals Notebook runs on your own private cloud instance. It's a possibility, but suffice to say, Signals Notebook standard itself is fully SOC 2 compliant. So if you have really if you have questions or concerns about that, we have one full documentation on what that means. I mean, SOC 2 compliance is really the standard for cloud security. And the simplest would also be to simply get in touch with a cloud security expert at Revity Signals.
Yeah, thank you, Pierre, for this answer. I believe the security which we have it at while since our primarily we have very secured cloud-based services. So yeah, thank you. So moving on to the other question, I would go to Ms. Rupali. Ma'am, we have basically got a question, especially based on your presentation, which says that what parameters should be factored while validating the ELN software as a part of CSV?
So as we discussed, so R&D data is to some part auditable data. by authority, and hence any digital application in R&D research labs also fall under GXP. The framework which is used to validate any GXP application holds true for any lab R&D lab applications as well. So GAM5 all validation criteria should hold true for research labs as well. But when you are doing certain OQ scripts, you can have the risk assessment. You can see what are the critical data and data fields. You can go for the regression testing for those fields. Certain repeated fields you can omit. You can do just once validation. So that is how you can define how can you reduce these OQ testing and define your test scripts and simplify it more. So that is how you can deal with it.
Yeah, thank you very much, ma'am, for your answer. And ma'am, there's another question which talks about the features available in Signal or the ELN to have a compliance dashboard with respect to Alconi Plus principles. So, would you like to share or would you like to answer this question also?
So I believe we discussed this that the design of ELN application has to be very strategic. So you have to have a fair balance between flexibility and the controls, where you can generate the data which is ALCOA compliant data. But I don't see that you can have a dashboard which can indicate whether your records and your processes that you have built in ELN can have this ready status on the ALCOA compliance. This is actually the expertise of the team and the SMEs who are going to build the application. So MSPR can also pitch in if he has anything in his mind to bring such kind of dashboard going forward, which can indicate that these workflows are ALCOA compliant.
Pierre, would you like to add something to it, or?
I apologize. I was not full. I was looking for the webpage for the SOC 2 compliance to give to as a question, so I did not fully pay attention. I apologize.
No worries, no worries. I will connect to the person who has asked this question offline, and we'll see if we can answer this question to the asker in a detail. Thank you. Mr. Rangarao, we had heard a great story. I mean, how your organization has taken a key decision to move to the Signal notebook. So I just wanted to know what was your biggest driver to move from the paper-based notebook to the digital platform?
It's a good question. I updated in the presentation, but monitoring CROs sitting remotely. We don't have a chemistry lab. Monitoring multiple projects, multiple series, and multiple CROs running parallelly, and maintaining them, following up, managing what's going on. So because it's a small biotech, if you are an MNC, you may not worry, but for a small biotech company like us, the productivity drives everything. So so that's where when we expanded our drug research from antimicrobial resistance to oncology, then we initiated multiple projects simultaneously from the scratch. So we expanded our chemistry resources also, and also the CROs. So that's where because we don't have much chemistry resources in-house. I'm the only chemist, and there is one more medicinal chemist and computation chemist. And pharmacists. That's all we have. So with a little resources, managing and monitoring multiple projects, multiple CROs is a big challenge. So that's where we took a decision immediately to look for an alternate solution which can solve all our problems.
Okay.
So Perkin-Elmer that time it fulfill all our checkbox, and that's why we took a decision immediately. And we also introduced very fast because I joined in 2020 August, and in October we already introduced in our platform. So it's very fast, and it requires a minimum 4 hours of training for the the end users, and 4 hours of training for the administrators. So it's very fast. Only thing is, if it is delayed, it delays from the CRO side because they have hierarchy to accept anything new. So it will delay that process. Otherwise, it will be very fast.
Okay. Thank you very much for this answer. Tanvira, can uh, uh, I was talking to a couple of users before this session also. And since Tanvira, are you with us?
Yes, I am.
Okay, thank you. Tanvira, you have shown the power of a signal notebook in a biosimilar research. And
Yes.
one of the user was basically asking me, can we visualize a biological sequence within the signal notebook?
Yes, the biological sequences, as I discussed within the workflow, and I showed that there is a dynamic view mode within which you can visualize the entire sequence. You also get to copy the sequences and the subsequences in certain formats, like in the inverse order. So yes, you can do that
Okay,
within
okay.
the
Thank you very
materials
much.
library. Yes.
Okay. Manish, are you with us?
Yes, Subir.
Yeah, Manish. I was talking to a couple of users before the session, and they have a question which says that a wide range of an experimental experiments are conducted in the formulation and the biology R&D, each requiring a unique set of data to be captured. How many experiment templates are available out of the box system?
Okay, so we believe the template should match your specific requirements and workflows. So system provides you infrastructure, it provides you platform to create templates. Uh, Templates can be configured without necessity of any coding. So the templates can be created with our services team, or you will be trained to configure it easily.
Okay, okay. Thank you very much, Manish, for this answer. Manish, there is one question from the audience also on the reagent library. The columns needed created are taken default from the materials library information. Would you like to share any insights on this?
Yeah, the libraries and the tables which you would like to see, that can be, of course, that can be configured. It depends on how you have configured, and definitely we can discuss this offline with the user if they are already using our system and if they have any specific questions. Okay.
Okay, yeah. Uh, I think that will be all. There are other questions also, and I believe those questions can be answered offline to the respective questioners who has asked these questions. So with this note, I would like to thank everyone once again for joining this India Connect event, and we really appreciate your time. And we will be sharing the recordings of this presentation, the entire session with you after this session. And we will definitely seek your feedback for our future sessions also. So that will be all from my side. Thank you very much.
Thank you. Thank you, sir. Yeah.
Thank you,
Thank you very
everyone.
much.
Thank you
Thanks.
Uh,
Thank you,
Thank
everyone.
you,
for
everyone.
joining.
Thank you for your time.
Have a great rest of your day.
Thank you.
Thank you, you too.